In yesterday lecture, we were discussing about the terms used in Mendel's experiment. Among which students we were going to discuss on the topics like dominant and recessive, phenotype, genotype, allele, allelomer, hybrid, hybridization, like the terms. Now students, the remaining terms will be discussed here on today. Let's start with the term monohybrid cross. Yes students, the term monohybrid cross. Students, in previous lecture I will define about the 7 selected traits of the Pisum sativum. It means sweet pea or green pea that was selected by the Mendel for his experiment. So students, in monohybrid cross, what is monohybrid cross? The monohybrid cross may be defined as the process of cross hybridization in which only one characteristics or traits is considered during the process of cross hybridization. Yes students, when only one of the contrasting characteristics is considered during the process of cross hybridization, such type of the cross is known as monohybrid cross. So students, let's understand monohybrid cross with the help of an example. When a cross is made by selecting or by considering the height of the plant, then what is the contrasting characteristics of the height of the plant? Yes students, tall and dwarf. Tall stem plant and dwarf stem plant, these are the two contrasting characteristics. What is the allele for the pure tall plant? Capital T, capital T. And students, what is the allele for the dwarf? Small t and small t. And when the process of cross hybridization is carried, then we find the result in the form of individual is capital T small t, capital T small t, here also capital T small t, and the last one is also capital T small t. Student, this is the parental generation. Yes students, this is called parental generation, RP generation in abbreviation form. Parental generation, and these are the alleles, the pure breed, homozygous type, capital T, small t, and the results that obtained after the process of cross hybridization. This is the first filial generation and called F1 generation. What is students? F1 generation, first filial generation. So students, what we obtain by the process of monohybrid cross hybridization, we obtain all the individual species. As a result, all the individual species are hybrid. Yes students, hybrid plants. In morphologically or phenotypically, all these individual plants are obtained are tall in height. It means their stem are tall. So students, such type of cross hybridization by considering a single kind of contrasting characteristics is said to be monohybrid cross. Let's see students, now we are going to discuss about the dihybrid cross. Yes students, the dihybrid cross, as its name clear that when we are going to considering the two pair of contrasting allele in the individual plants, then such type of the cross hybridization, this is called the dihybrid cross. Let's see on dihybrid cross. Dihybrid cross. Students, I am going to take the example to define the dihybrid cross quite well. Then students, here I am going to take the two contrasting pair of allele, and after that we make the process of cross hybridization with the help of a chart. This is called Punnett chart. Yes students, Punnett was the biologist or scientist who gave first multiplication bonds for the process of cross hybridization. Hence, after the name of him, this is also called Punnett chart. So students, when two pair of contrasting traits are considering for the process of cross pollination, or cross hybridization, then this type of the cross hybridization is said to be dihybrid cross. So students, here I am going to take two pair of contrasting characteristics from the Pisum sativum, it means the sweet pea or garden pea. First I take the height of stem. What are the contrasting traits? The dominant is tall and its area is capital T, whereas the recessive is dwarf and their area is small t. Next student, here I am going to take the second contrasting characteristics or trait is the flower color. Yes student, flower color. Then here I am take red as the dominant trait and representing this by capital R by the allele, and for white flower here the allele is smaller and smaller. So students, when the process of cross hybridization made between these plants by considering both contrasting pair of allele, then the individuals are obtained always in 9:3:1 by genotypically. This is the genotypically ratio. So students, What are the key terms for these contrasting characteristics? The key terms for the process of cross hybridization from capital T, capital R, small d, and small r will be capital T, capital R, capital T, small r, small d, capital R, and small t, small r. These are the possible alleles that undergoes in individuals. Now here I am going to draw the Punnett chart, whose about I will tell you. This is the multiplication box and called Punnett chart. This is the symbol of cross hybridization, and here I am going to draw the 4 boxes. Horizontally and also vertically. So students, there will be 16 boxes we obtain by making horizontal and vertical rows and columns. Now I am representing the allele. The key terms are key alleles: capital T, capital R, capital T, capital R also, capital T, small r, capital T, small r, small t, capital R, also small t, capital R, and small t, small r, and here also small t, small r. Now students, we are crossing these pair of contrasting allele, not mathematically but biologically. So students, when the process of cross hybridization between these alleles, then the results is obtained. Look here very carefully. Capital T, not T square. It is not calculated as mathematically. Capital T, capital R, capital T, capital R, small r. The capital letter is always written ahead, and when small comes, then written after that, because capital is the dominant trait and small is the recessive trait. Okay students, capital T, small r, capital R, capital T, small t, capital R, small r. Now here the capital T, capital R, small r, capital T, small r, capital T, small t, capital R, small r, capital T, small r. Now here capital T, small t, capital R, capital T, small t, capital R, small r, the small t, capital R, and students, the small t, capital R, small r. Now here capital T, small t, capital R, small r, capital T, small t, and small r. This will be small t, capital R, small r, and this is the small t, and small r. This is the pure recessive trait. Now students, how we calculate the genotypically ratio? The results should be calculated here by referencing them as tall red. This is also tall red. If one of the dominant trait is present, then this overlaps the recessive one. So students, these are the tall red, the tall red, tall and white, because both of the alleles are of recessive types, hence it comes in appearance as white color of the flower, tall red, tall white, the tall red, tall red. Here students, both of the contrasting alleles are small t, hence this is the dwarf red. This is also dwarf red. This is the tall red, the tall white, dwarf red, and this will be dwarf white. This is the only single individual that is the dwarf and white. So students, the results should be calculated genotypically. How we can calculate? The tall red ratio tall white ratio dwarf red ratio dwarf white. Okay students, then calculate tall red 1, 2, 3, 4, 5, 6, 7, 8, and here 9. These are the 9 tall red individual species are obtained, and tall white 1, 2, the 3. These are the 3 tall white individual species are obtained in F1 generation, in F2 generation. This is the second serial generation, because first I will cross here by the key terms, and these are the individuals of F2 generation. Now students, dwarf red, the dwarf red 1, 2, and this is the 3 dwarf red, and dwarf white, this is the single dwarf white. So students, the genotypical result is obtained 9:3:3:1. Hence students, this is called the dihybrid cross. Now students, we are going to discussing another topic. This is the back cross. Back cross. Students, what is back cross? When the cross made between the parent and the individuals of a whole generation, such type of cross is said to be back cross. These are the very short topics. Then the parental generation, here I will take pure tau as example. As its name clear that this is the back cross, the cross made between the parental generation and the individuals of F1 generation. Yes students, here I will take the individuals of F1 generation that are the hybrid tau by taking the alleles for the height of the plant stem. Then after cross hybridization, we obtain the result capital T, small t, and here capital T, and capital T small. So students, what we find the result? The result we find genotypically the 2:2 are in simple fractional ratio 1:1. It means 50-50%. Genotypically 50-50% ratio is obtained when the cross is made between a parental generation and the individuals of F1 generation. This is the F2 generation. Okay students, F2 generation. Likewise, here we can also make the cross hybridization between a pure dwarf, and it means dwarf, the parental generations, small t and F1 generation hybrid top, capital T small t, the capital T small t, small t small t, capital T small t, and the small t small t. Here also student, the same result is obtained genotypically. The two individual species are hybrid top. The result of hybridization genotypically hybrid dog ratio dwarf. The 2:2 are in simple fractional ratio 1:1 are 50-50% chances of having the hybrid are the dwarf species of the plant. And the last of the topic of the mental step, that is the bad test cross students. What? The test cross. This is the last topic, test cross. So students, what is test cross? The cross made between the individuals of F1 generation. Yes students, when the cross made between the individuals of F1 generation, it means hybrid